or solid phase reversible immobilization beads are a huge help for people preparing libraries for next generation sequencing using a short read technique like Illumina. Basically, if you want to sequence RNA or DNA with these techniques, you start by preparing a library where basically you shear up or you fragment or you somehow make into smaller pieces the DNA or the RNA. Then there are a bunch of steps where you add adapters, you do some PCR in order to make more copies so you have a higher concentration going into the reader. And if you're doing it from RNA, you're also going to have a reverse transcription step where you make DNA copy of that RNA. So lots of various steps where there's lots of places that you're adding in things. And at the end, you want this really pure DNA that you're going to go get sequenced. And so you need to have purification steps along the way and at the end to get rid of all those things like adapters and primers and salts and enzymes and all these various things. And thankfully, these beads make it really easy because they allow you to bind nucleic acids to these beads, use a magnet to capture those beads, and then wash that other stuff off, and then elute or kind of like unbind your nucleic acids from without all that other stuff present. What's really cool about these beads is that they allow for size selection. So basically, depending on the ratio of beads to your sample, you will get different sizes of nucleic acid selectively bound, and this is going to allow you to choose kind of like a size range that you want to keep, get rid of really big stuff that wasn't sheared very well, maybe it's not going to read well, or get rid of really small stuff like those primers, like those adapters, like those adapter dimers and things like this. So if you're like me, you're wondering how this works. If you're really like me, you look it up, and if you're really, really like me, then you tell other people what you learned. And so here goes.
So commonly, these beads are going to be used when we're doing this step where we're basically adding the adapters and maybe doing some pre-amplification of these sequences. So we take this DNA, we have these smaller pieces, we add these adapters. These adapters are going to have a barcode. They're going to have something that's going to allow them to bind to this chip, the sequences on the chip. And then they're going to, lots and lots of copies of them are going to be made on this chip. And then those are going to be read out as they're getting made. It's called like sequencing by synthesis. And then you can take all those short reads and line them up. So it's really cool. But in order to get all of this, you have to add those adapters, maybe do some pre-amplification, things like this, various steps where we're going to want to remove things like primers and extra adapters, adapter dimers, things like this. And so these beads are going to come in really helpful because they're going to allow us to do this in a potentially like high throughput manner without having to do any sort of gels for size selection, without having to do any sort of those like spin columns or things like this. We can just do it directly on, say, like a PCR rack or even in a plate with the high throughput, really high throughput manners. So how it's going to work is we're going to take that fragmented DNA or RNA, and there are different methods that you can fragment it. Some of them use like mechanical shearing, some of them use chemicals, but you want to break it up into those smaller pieces because those pieces are going to be what the Illumina is going to be able to sequence. And then we're going to want to be able to basically enrich for the pieces that are the right sizes. So we're going to have to get rid of those small things such as the primers and the adapters, and you can get weird things like adapter dimers, and we want to get rid of those, which we can do during using the beads. And also if we want, we can get rid of the bigger things, and we'll talk more about how we do that later. But first, let's talk about what's actually going on. So first off, what are these beads? These beads are brand names for them include like SPRIselect, AMPure, NEB sample purification beads. There's lots of different names. Basically, they are these little beads, and on the surface of them, they have like a magnetic coating, a paramagnetic coating. So it's paramagnetic. Basically, it's acts like just like a normal molecule, like they're not all sticking together to one another. You have this solution. But then if you place them on a magnet, they're going to become magnetic and kind of like cluster against the surface of the magnet, which is going to allow us to take things off. So you can see that it's all getting drawn towards the magnet. And this is going to allow us to then isolate the beads from the liquid around them. And the beads are either going to have our DNA or our RNA bound, or they're not, and depending on what the liquid is. And so we'll use wash conditions where they don't bind, and then we're going to elute them under conditions where they bind, and we'll get more into this. So these beads are really really helpful, and they're going to allow us to do things easier than if we had to use spin columns or we had to use those gels. But really, the real magic comes not from those paramagnetic beads, no matter how cool they are, but actually from the solution that they're in. They're in a solution of PEG and sodium chloride. And so PEG is this long molecule that we'll talk about later, but it's kind of big and floppy, and it's going to crowd out the DNA, and it's also going to take up a lot of water. And so it's going to help promote the precipitation and the kind of clumping together of your DNA or your RNA onto the beads. In terms of the solution that these actually are in, it's typically like 20% PEG, 2.5 molar sodium chloride, which is a lot of salt. And so as we'll talk about later, both the salt and the sodium and the PEG are going to kind of hog a lot of water. The salt's also going to neutralize the negative charge of the backbones, as well as the charge of the carboxyl groups on the surface of these beads so that the DNA or the RNA can bind. There are different solutions that are used for DNA or RNA. So these beads, I should say, are really expensive. I found this basically do-it-yourself guide to make cheaper version. You can't buy you have to buy the beads themselves, but the solution that they're in, you can make this solution yourself and save some money. And so you can see that in addition to the PEG and the sodium chloride, there's also Tween, which is a detergent, a pH buffer, so tris or trisodium citrates, things like this, and different conditions for DNA versus RNA, and that's all. I think that the beads you can use the same beads, they just have different buffer conditions, and they both have that same high sodium chloride and high PEG. The PEG 8000, basically, there are different molecular weight PEGs that have different lengths, and so the PEG 8000 is what's typically used. So we have the solution of PEG and sodium chloride, and this is going to promote the binding, the precipitation, and the binding of those nucleic acids to those beads. What's really cool, as we'll get into, is by modifying the beads, this ratio of the bead solution, so the solution of the beads with the PEG and the sodium chloride, to the amount of the solution containing your DNA or RNA, you can selectively bind sizes of interest. So basically, when we're talking about these ratios, what we're really talking about is what is the concentration going to be of the beads and the salt. So at a higher concentration, at a higher ratio of the beads to the solution, you're going to have a higher concentration of these, and at a lower ratio, you're going to have a lower concentration of these. And that's going to determine what does or doesn't bind, because as we'll talk about, the bigger things are going to bind more easily, so they're going to bind at a lower ratio, whereas it's going to take a higher ratio in order to bind the smaller things. Once they're bound, we can use that magnet to capture the beads against the surface, and now we want to pipe out the other stuff. So at this point, our DNA or RNA of interest is going to be bound to the beads. When we're talking about a typical one, so when we're talking about your general size selection, we're not talking at this point about the double size selection, where at this point our DNA of not interest would be bound. At this case, in most cases, your DNA of interest is going to be bound at this point, and so you can then use the magnet to capture the beads and pipette out the other stuff. And so that other stuff is going to be like your salt, your PEG, and small pieces of nucleic acids. So as I mentioned, and as we'll get much more into, the smaller things are going to be harder to precipitate, and so you're going to need a higher ratio. And so at a lower ratio, you're not going to precipitate out those small pieces. And so this is how we can remove those primers and we can remove those adapters and things like this. So the DNA or the RNA is bound to the beads and the beads are bound to the magnet. Well, now you can go in with your pipette and you can pipette out the liquid. Now what you're going to do is you're going to wash them with ethanol while they're bound. So typically like 80% ethanol. Under these conditions, the DNA or the RNA is going to stay bound, but those salts and stuff, they can still come off. And so you're able to wash that stuff off. Typically you do like 2 washes. And then you make sure you get rid of all the ethanol, you dry the beads out, but not totally, so that you can still redissolve them. And when you redissolve them, you add a water or you add an aqueous buffer such as TE. And this is you're going to then allow it to redissolve. And now that they're redissolved, you can take that solution. So now you take that solution that now has your DNA or your RNA, and you're going to keep this. But how does it actually work? Basically, the salt and the PEG hog the water, leaving less for DNA and RNA. So when we talk about something being dissolved, it means that each copy has a full coat of water. And DNA or RNA, they're like coated in water typically. They have this solvent layer, this tight layer of water all around them. And the water really likes hanging out with their backbones because those backbones are negatively charged. And so if we want to precipitate it, we want it to come out of solution, we're going to have to remove all of that water and make it so that the water doesn't want to hang out with it as much. And one way we can kind of like remove water is by making it seem like there's less water in rounds, by having other things take up the water. And so when you have those salts and when you have those PEG, well, they can bind to the water too, and they're going to hog it so that it seems like there's less water in the solution because there's less free water available to bind to your nucleic acids. Additionally, PEG is going to cause a crowding that promotes the DNA or the RNA sticking together. So PEG is this big floppy thing, and we use it actually for like crystallization when we're trying to get proteins to bind to one another because it crowds things out. In addition to taking all that water, it basically wiggles all over the place and takes a lot of space, leaving less space for your DNA or for your RNA. Additionally, those salt ions that you had, they're going to shield the charges. So when you put the sodium chloride you dissolve it, it dissociates. So it breaks up into sodium ions and chloride ions. And those sodium ions are positively charged, and they're going to coat the negatively charged backbone. And this is going to allow it so that the DNA and the RNA strands don't just repel one another, and so that they don't repel the carboxyl groups that are on the beads. So when we talk about carboxyl groups, you should be thinking, oh, those are normally negatively charged, right? And yes, these are going to be negatively charged. And this is going to allow them to kind of make this process, this binding, reversible. It's going to help make this reversible. So you might have heard of methods like using biotin tags to bind to streptavidin to purify things out. Well, that's going to be a really, really strong interaction. And it's going to be hard to reverse. But in the case of this DNA or this RNA binding to this beads, this is going to be easily reversible. And part of the reason why it's easily reversible is because you have those carboxyl groups that are going to then repel the DNA or the RNA when the conditions are more normal, and when we're not under those conditions that are promoting the precipitation. And so the salt ions are going to help kind of bridge between the carboxyl groups and the DNA or the RNA, shielding their charges from one another and allowing them to bind. So putting it all together, you take that fragmented DNA or RNA, and now you're going to add it to the beads with the PEG and the sodium chloride. Those are going to hog up the water. the PEG is going to flop all over the place and make it hard for these molecules to swim around, and instead promote them kind of grouping together to one another and to those beads. So they're going to precipitate and bind to the beads. You can then capture those beads with a magnet, pipette off the liquid containing all that extra salt and PEG and things like this. Wash with ethanol while the DNA or the RNA is still bound, and then make the conditions for them to unbind or elute. And so you can add this aqueous buffer and then they'll come off, and then you just pipette that out and you've got your purified DNA or your RNA. So now let's talk a little bit more about how we can modify the bead solution to sample ratio to selectively bind sizes of interest. And so remember, when we're talking about this ratio, what we're really talking about is kind of like the concentrations of the sodium chloride and the PEG compared in the solution. So the higher this this ratio is, the smaller the DNA or the RNA that will precipitate and bind beads. So it's going to be easier to precipitate out the bigger things. Each of those bigger DNA or RNA fragments, to be dissolved, and remember, it has to have a full water coat. Each copy has to have a full water coat. And when you have these big molecules, they need a lot more water to coat them. Additionally, once they are like uncoated, once they precipitate, they're going to form a lot of interactions with one another. And so there's lots of places where they can glob together, which means that there are lots of interactions that then have to be broken in order for them to redissolve. So it's going to be easier for them to precipitate and stay precipitated and bind to these beads. So even if you have a lower ratio, something like a 0.4, I think around 0.4 is basically the lowest it'll precipitate out at. A ratio of 0.4 of the bead solution to your sample solution. Now you're going to only precipitate and bind the big pieces. And so if you were to look and say, or you take your sample and take what was bound and go stick it on some sort of analyzer, if you had the size of the fragments over here and the amount, so the signal you have over here, so you can see that you'd be kind of choosing things, only keeping things that are in this big range. Well, what happens now is if you raise the ratio, well, now you start selecting for the medium-sized ones as well. So you still have those big ones, and now you're starting to have the medium-sized ones too. And if you go even higher of a ratio, you start to bind to the small pieces as well. And according to literature and stuff, like, you also get lower efficiency of binding to those bigger things when you have a higher ratio. And so you'll see that the peak is actually going to get shorter too. So you're not getting quite as much of the big stuff dissolved. And so here are some actual traces from this paper, Improved Protocols for Illumina Sequencing. And you can see that as you decrease this ratio, you're getting smaller and smaller things. And I will post a link to this as well. So that's great. If we use a higher ratio, we can exclude those smaller things like adapters and primers. But what if we want to exclude bigger things as well? Well, you can do something called a double-sided size selection, where you start by doing a low ratio to remove the large stuff by getting rid of the beads, and then follow that by using a high ratio on the supernatants. So you take the supernatants after you discarded those beads, and now you do another purification, but this time you're going to do it with a higher ratio. And then this is going to then allow you to select for those smaller things. But by kind of making it not too high, you can still get rid of some of these really small things. So basically, it works on that principle we've been talking about, how it's going to be easier to precipitate out those big things. So we're going to start by getting rid of those using a low ratio. So we don't want to go too high, because then we're going to be binding to the smaller things as well. And at this point, we're going to actually want to get rid of the stuff that's bound. So we're only going to want to bind the really big things. We then use a magnet to isolate those beads. And in this case, we're going to get rid of those beads. So normally we're going to keep the stuff that's bound, but at this point, we're going to get rid of the stuff that's bound. And we then take the supernatant, so the stuff that didn't bind, and then we're going to add it to more of the beads and PEG and sodium chloride mixture. And now we have smaller pieces, but not tiny ones bind too. So you choose the percentage or you choose the ratio so that the things that are really small aren't going to bind, but the things that are the size you want will bind. And you've gotten rid of the big stuff. And so now you should be left with mid-sized pieces. So what does it actually look like? Here's a user guide for one of these types of beads, this SPRIselect from Beckman Coulter. Not a paid endorsement or anything. They just have a nice user manual, and these beads are commonly used. So these are like the beads that are used in the NEB sample purification beads or whatever. So they have a couple of different names for the two selection steps. So that first selection step that you do is in the double-sided is what we call a right size selection, because basically what you're doing is you're selecting the cutoff for the right side, the cutoff for the big things. So remember, when we have this first step, we're going to be removing the big things, and we're going to be getting rid of those beads. And so when we talk about setting the right side limit, this is okay. So what do we want to keep? What is the smallest of the big things that we want to keep? And by increasing the ratio in this first selection, so this right-sided selection, we're going to exclude smaller big things. So remember that the higher the ratio, the smaller the things that can bind. And in that first step with the double-sided, we're getting rid of those things. So by increasing the ratio in this first step, you're then narrowing it on the right side. Now, what about the left-sided selection? So this is like what we normally do, is typically a left-sided size selection. We're setting the lower limits. And so we're saying, okay, if we go too high, we're going to bind the smaller things. And in the left-sided size selection, we're keeping what's bound. And so if we have a higher ratio, we're going to be binding smaller things, and we're going to have this kind of like, our peak is going to shift out to the left more. So you can see with a higher ratio, you're going to have more of the small stuff included. Whereas if you were to lower that ratio, well, now you're not going to include those things. And so you're going to, when you lower the ratio in the second selection, this left-sided selection, you're going to narrow the left-sided cutoff. And so when you can change both of these ratios in order to narrow down on the size of interest. And so you can choose the ratios you want based on the sizes that you want. And so typically you're choosing sizes somewhere in the hundreds range, a couple hundred to up to like 1,300, I think, if you go to a low ratio. If you want bigger size things, then you might have to modify things. And so here's an article that's kind of playing around with different PEG concentrations and different salt concentrations in order to precipitate out those bigger pieces. And so I will post a link to this as well, where they kind of play around with that. For normal things, though, Illumina has some nice resources, including this guide to help you do the calculations for what volumes of these different solutions you need. And Eric Chow from UCSF, he has this great these iBiology videos that do a good job of explaining the process of preparing a sequencing library, as well as a practical video on how you can use these magnetic beads. And finally, I just want to show you guys where this all came from. So here's this 1975 paper that kind of lays the groundwork for this. John Liss and Robert Schleif, they're basically showing that the PEG and sodium chloride is able to precipitate DNA in a size selective manner. And what they're doing, though, is they're not using beads at this point. They're just kind of like centrifuging things down. But they show that as you increase the concentration of PEG, well, now you're going to be keeping smaller and smaller pieces, just like we talked about. And they also do that sequential thing. They do this sequential thing where they take the supernamed and then they add higher amounts of PEG, and they keep doing this over and over. This is like what we were talking about, how you can size select different ranges. What's kind of confusing in this paper is I think that they mislabeled their figures, figure 1 versus figure 2, but hopefully you can make sense of it. Additionally, so that was just, remember, that wasn't using the beads, but then the beads started being used in like 1995. So this is technology's been around for a while. And here's this nucleic acids research paper from 1995, DeAngelis, Wang, and Hawkins, where they showed that, yeah, you can do this on these beads, and it's really, really helpful because then we don't have to do all that centrifuging, and we can do the size selection without having to do gels, all this cool stuff. And since then, there's been further improvements and things like this. And so I'll post links to lots of these great resources. But yeah, it's a really cool technology, and it was really fun to learn about how it works.